Hello students, welcome to the EPG Pathshala. I am Dr. Hani from University Institute of Pharmaceutical Sciences and Research from Baba Farid University of Health Sciences, Faridkot, Punjab. Today we will discuss about this module Targeted Drug Delivery Systems Part 2 under the paper Novel Drug Delivery Systems Part 2. The learning objectives of this module will comprise of active drug targeting and passive drug targeting. So let's start with the introduction. In order to design a robust drug delivery system, it is quite imperative to comprehend the tumor environment and that there are various modes through which the drug can be targeted. The types can be classified broadly into the following: active type of the targeting, passive type of the drug targeting, physical type of drug targeting, dual mode of drug targeting, double drug targeting, inverse drug targeting, combination drug targeting, and chemical drug targeting. Let's start with the concept of the active drug targeting. The term active targeting has a flavor of guiding a drug or a drug carrier to a targeted site like a cruise missile does. Currently, the drug delivery systems do not have the ability to guide themselves to reach a specific target. Active targeting is basically a specific interaction between the drug or drug carrier and the targeted cell with the help of a specific ligand-receptor interactions. Generally, a ligand or an antibody is incorporated for active targeting against a specific surface receptor. For example, epidermal growth factor receptor known as EGFR, transferrin receptor TFR, and folate receptors FR. These receptors or the antigens, they are usually overexpressed on the different malignant cancer cells. So ligand receptor interactions, they are possible only when the two components, that is the ligand as well as the receptor of the targeted cell surface, they are conformally fit with each other at a particular area or the close proximity of a less than 0.5 nanometer. In this particular figure, you can see a nanoparticle in which the receptor of the targeted cell where the drug has to be reached, there is an interaction between the ligand as well as the receptor. So this particular ligand receptor mechanism is the principle of active drug targeting. So students, let's start with the active drug targeting. The active drug targeting is basically a ligand receptor interaction mechanism which only takes place in a close proximity of 0.5 nanometer. Conventionally, the drug delivery systems reach to the targeted site as a result of. In this particular slide, we will discuss the various ligands which are used for the active drug targeting. The examples of some of the ligands which are used for active drug targeting are as follows: antibodies and their fragments. The example of such antibodies is the Fab and SCFV. Antibodies and their fragments, they are extracted and designed through biochemical and genetic recombinant techniques. There are two antigen-specific fragments of the antibodies which act as ligands on various tumor cells. These include antigen binding fragments, which is Fab, and a single chain variable fragment, which is SCFV. In this particular figure, you can see the illustration of the specific antibodies and their fragments in comparison to their conventional antibodies. Another example of the ligands which are used for the drug targeting, they are the proteins. These ligands are the endogenous proteins which selectively bind to a specific membrane-bound receptors which are present on the cells. However, the limitations of such ligands are the immunogenic reactions and the offset target adverse effects. Another example of the peptides which also act as the ligands. These ligands, they exhibit a small size and relatively have a lower immunogenic potential in comparison to the proteins. The another advantages include better stability and ease of conjugation onto a nanocarrier surfaces. The examples of peptide ligands include cell-penetrating peptides such as TAT peptides, RGD peptides, which are expressed on the tumor cells. In this particular slide, we described the various ligands as we describe the peptide as the ligands for the active drug targeting. The example of peptides are the cell-penetrating peptides such as TAT peptides, RGD peptides, which have the amino acid sequences of arginine, glycine and aspartame. So in this figure, you can see the overview of the carrier ligand interaction between a receptor and the penetration of the peptide into the cell. The cargo vesicles that attach to a particular receptor present on the tumor cell in a blood vessel, and this particular carrier or the cargo system is further penetrate into the tumor cell. The example are the IRGD peptide as well as the neurophilin, which is the receptor present on the tumor cell. In this particular slide, we will study various receptors for the active drug targeting. The descriptions about some of the important receptors which are widely exploited in the targeted therapy, they are transferrin receptor, folate receptor, integrin receptor, human epidermal receptor. Let's start first with the transferrin receptor. It is the fourth most abundant serum non-heme iron binding glycoprotein which is highly upregulated on the surface of many cancer types and is also efficiently internalized. Hence, it may act as a prognostic cell marker for the specific tumor recognition and is also a striking receptor for the targeted cancer therapy. The second receptor, which is the folate receptor, this receptor, which has the molecular weight of 441 Daltons, is a member of B complex protein of vitamins, is a vital cofactor in carbon transfer reactions for the biosynthesis of nucleotide bases. This receptor plays a vital role in cellular functions, nucleotide synthesis, epigenetic processes, as well as cellular proliferation. The major advantage of folate receptor is its stability, cost effectiveness, non-toxicity, as well as non-immunogenicity. Further, folate receptor is also internalized by the receptor-mediated endocytosis process. The next receptor is the integrin receptor. These receptors, they are the heterodimeric cell surface receptors which are overexpressed on the tumor-associated endothelial cells. And these play an important role in the calcium-dependent signaling pathway leading to the endothelial cell migration. Further, these receptors represent a potential pharmacological target for the anti-angiogenic therapy. The next receptor is the human epidermal receptor. In this particular receptor belongs to an enzyme family of tyrosine kinases which contributes significantly in the cell proliferation, differentiation and cell growth. The family consists of the four members, which is EGFR or also known as HER1, HER2, HER3 and HER4. So this particular receptor is overexpressed in wide range of tumors such as breast, cancer, lung cancer, colorectal cancer, and pancreatic cancer. So students, today I am going to tell you a successful story of the most promising actively targeted nanoformulation, which is codenamed as Bind-014. Docetaxel loaded nanoparticles by conjugating with SS235 amino 1 carboxypentyl uridopentoic acid, which is also known as ACUPA, were actively targeted to a prostate specific membrane antigen, also known as PSMA. The targeted system based on this particular antigen, which is ACUPA, a prostate cancer specific imaging agent, resulted in the various commercial products based on the active drug targeting. As such, there are no actively targeted nanoformulations which are available presently in the market in the commercial form. However, about 5 targeted liposomal formulations and 2 targeted polymeric nanoparticle therapeutics have made into the clinical investigation developmental phases. The example of such products include MBP426, MCC465, SGT53, MM302, Bind-014, CALAA01, cetuximab, decorated doxil and calyx liposomal formulations. So such commercial products, they are only in the clinical phase and no commercial products are available currently in the market. Targeted drug delivery systems, they have found an immense application as an ideal approach for the drug delivery to the brain. As active targeting is a non-invasive approach, the drug delivery to the brain after crossing the blood-brain barrier can be exploited through various influx transport mechanisms such as cerebral endothelial, including the carrier-mediated transport, receptor-mediated endocytosis, and adsorptive-mediated endocytosis. These are the processes which are exploited for the transport of a drug into the blood-brain barrier. So various technologies such as liposomes, nanoparticles, dendrimers, they are being investigated for the brain pathologies. The blood-brain barrier is basically a specialized network of capillary endothelial cells which is partially covered by the pericytes, basal membrane, and surrounded by the end feet of the astrocytes. So this particular barrier acts as a key challenge for the delivery of the drugs to the brain tumors. In this particular figure, you can see the blood-brain barrier across the endothelial cell and as well as the cell junctions which are present on the blood. And the endothelial cells, they are enclosed by the pericyte membrane as well as the astrocytes. So the transport of the drug across the blood-brain barrier need for the vesicular transcytosis for the internalization process in case of direct brain administration. So this particular technology, so about 98% of the small molecules or the genes and 100% of the large molecules such as proteins, they are prevented to reach to the tumor sites in the brain. Further, the drug transport to the brain is limited due to the presence of tight junctions, metabolic enzymes, and immunological barriers. Active targeted approaches such as absorptive mediated transcytosis, transported mediated transcytosis, and receptor mediated endocytosis have been utilized to deliver the drug to the various sites of the brain. Active drug targeting to the tumor site. The preference of the active mode of drug targeting over the passive targeting is due to high affinity of the ligands toward the receptors on the tumor cell which are overexpressed in comparison to normal tissues. The surface chemistry of the nanocarriers will decide the target recognition and binding to the cell through ligand-receptor interaction mechanism. Further, the efficiency of the active drug targeting also depends on the homogeneous expression of the receptors, which further lead to internalization of the nanocarriers via receptor-mediated endocytosis. In this particular slide, we will present a case study of doxorubicin targeting using a cyclodextrin-based nanoparticle system. In this system, a strong affinity towards the folate receptor has been presented. The hydrophilic folic acid conjugated cyclodextrin complex was designed, and the doxorubicin entrapped adamantane complexes, they were further conjugated with the folic acid based cyclodextrin complex using a mixed solvent precipitation method. It was observed that the drug was delivered to the cancer cells specifically than the non-cancerous tissue, and the targeting of the drug was found to be 8-fold increased to the specific target site via the receptor mediated endocytosis. So this particular study showed a promising case of the active drug targeting via the ligand receptor mechanism as shown in this particular figure. So students, we will discuss about the passive drug targeting, which is also known as spontaneous drug targeting. Passive drug targeting is defined as a mechanism to augment the target versus non-target ratio of the delivered drugs by minimizing non-specific interactions with the non-target cells without the influence of energy. The passive approaches which are targeted to the system. In this particular section, we will learn about the various clearance systems like reticuloendothelial system, which acts as a resistance barrier for the passive drug targeting. It has been found that nanocarriers, they are taken up by this particular system, which is reticuloendothelial system, because of the surface properties of the nanocarriers. Hydrophobic carriers, they are more prone to be cleared by the RES system in an order of liver, spleen and lungs. That is, liver is the major site in which RES clears the hydrophobic carriers in comparison to spleen and lungs. Further, the high circulation time and specific targeting as well as the size of the nanosystem should be within the 100 nanometer for the nanocarriers. The carrier systems should also be equipped with the stealth properties by using the polymeric coatings, which are hydrophilic based coatings made up of polyethylene glycols, poloxamines and poloxamers. Enhanced permeation and retention effect, also known as EPR effect. EPR effect is a phenomenon due to its unique anatomical and pathophysiological characteristics of the tissues. For example, majority of the solid tumors, they show a high vascular density or hypervasculature, or also known as angiogenesis. That is one of the most important features of the tumors to sustain their rapid growth, in contrast to the normal tissues and other organs. It is a pertinent fact that under certain pathological states like tumors, infarcts, inflammations, or the permeability of the vascular endothelial increases, then the tissue or the tumor become leaky and contributes to the passive accumulation of the drug or drug nanocarriers. This particular effect is also known as EPR effect. Large amount of the preclinical and clinical studies have highlighted the EPR effect as a complex association of various processes such as angiogenesis, vascular permeability, hemodynamic regulation, genetic heterogeneity among the tumors, and lymphoangiogenesis and heterogeneous tumor microenvironment. In this particular image, you can see that a tumor site in which the internalization of the nanoparticle occur through the receptor-mediated endocytosis via active targeting. While in case of passive targeting, the endothelial cells, they freely move in the systemic circulation and they get accumulated and diffuse at a particular site in the tumor tissue because of the leaky and highly permeable vasculature of the tumors. The basic mechanism of the EPR based strategy also shows that the pore size of the blood vessels in the healthy tissues is about 2 to 6 nanometer. Whereas in case of tumor tissues, the pore size of the blood vessels increases from 100 nanometer to 780 nanometer. Hence, the size of the drug particle which has to be chosen for the targeted system should be in accordance with this particular range according to the EPR mechanism. The application of the EPR based strategy for designing of the tumor targeted approaches, it is observed that the EPR based strategies requires the macromolecular drugs to have a longer circulation half-life and a sufficient effective pharmacodynamic level. This slide shows the techniques for the augmentation of EPR effect. In order to improve the design of EPR based tumor targeting strategy, that is the macromolecular therapy, various novel targeted systems have been developed through polymer modification. This modification leads to 10 to 200 times higher concentration in the tumor than that of normal tissues, such as skin, muscle, heart, kidney, and after systemic administration. The surface of the drug or the drug nanocarriers, they are functionalized using a water-soluble polymers with a well-solvated and flexible main chain such as polyethylene glycol (PEG) or styrene malic acid (SMA), N2-hydroxypropyl methacrylamide (HPMA). In this particular figure, you can see a pegylated molecule which acts as a nanocarrier, and the pegylation has been carried out onto the surface of a nanosystem which helps in the enhancement of tumor concentration through EPR mechanism. Indicates the process of pegylation. As you can see in the figure number 10 and 11, there is a functionalized polymer which is a targeting ligand in the form of red dots, and the green dot shows the therapeutic drug, and the chains, they are the PLGA and PEG polymeric chains. So the functionalization through the pegylation process starts with when a chain made up of PLGA and PEG polymer, when interacts with the therapeutic drug, then it leads to the formation of a nanocarrier. Then the nanocarrier is further conjugated with the targeting ligand so that the cellular recognition of the nanocarrier can take place. There is another technique which is also used to enhance the EPR effect. The use of vascular mediators such as nitric oxide, bradykinin, vascular endothelial growth factor, all such vascular mediators, they are used for increasing the permeability of the tumor vasculature to enhance the EPR effect. In this case, the EPR effect is achieved by the use of vascular mediators, which helps in the better drug accumulation at the targeted site or the tumor site. Example of the commercial products which are based on the EPR based passive drug targeting. The EPR based effect has emerged as a gold standard for the anti-cancer drug designing. A marketed formulation known as Doxil, which is a doxorubicin incorporated into the long circulating polyethylene glycol coated liposomes, is one of the excellent example of EPR based drug targeting. The principal factors affecting the EPR effect includes vessel architecture, interstitial fluid composition, extracellular matrix composition, phagocyte infiltration, presence of necrotic domains, factors pertaining to the colloidal carriers such as blood circulation time, particle size, particle shape, surface charge, and surface modification, or the pegylation process. Some of the other distinctive factors which also affect the EPR effect in the solid tumors are the extravasation of the colloids from the blood vessels, diffusion through the extravascular tissues, and the interaction with the intracellular as well as the extracellular targets within the tumor microenvironment. Although the targeting of the drug component is localized for the specific targeted cells, however, most of the targeted cells in the organs or the tissue, they are extravascular and are not bathed in the blood but in the extravascular fluid. The first aspect, which is extravasation, implies that if the drug or the drug carrier is in the bloodstream, that is either by absorption from the gut or through the IV administration, it can only reach the targeted cells by passing across the walls of the small blood vessels and entering the local tissue fluid. This process is called as extravasation. The two factors, namely extravasation and diffusion from the extravascular tissue, they are due to the diffusive and the connective forces which are influenced concurrently by the tumor biology and the characteristics of the diffusing species. The third factor, which is the interaction mechanism with the tumor microenvironment, is poorly understood. The interaction behavior of the colloids with the tumor further depends on the nature of the material, its affinity for all the components of the tissue, and the tumor composition. Here in this slide, we will discuss the various challenges or the limitations of the EPR based passive tumor targeting. Due to the high interstitial fluid pressure, it has been observed that only a fraction of the drug, which is about 4% of the total administration dose, reaches to the targeted site or either the tumor site. The EPR effect is also not reliable except for the primary tumors such as in the kidney as well as in the liver. The blood vessel density, distribution, and the leakiness of the solid tumors, they are also not practically known. The transport barriers in the extracellular matrix of the clinical tumor, they are also not measurable before the treatment. Passive targeted nanoformulations may end up in the release into the drug of passive targeted nanoformulations may end up in release the drug into the tumor milieu rather than within the cancer cells. This phenomena is known as PEG dilemma. Those drugs which are not readily absorbed or released outside the tumor cell, such systems, they are not able to achieve the effective concentration over an extended period of time. Now we come to the summary and the conclusion of this particular module. Scientists are frequently using the EPR effect and pegylation for the longer systemic circulation profiles and the higher drug accumulation at the targeted area of the tumor. But the reality is, the actual percentage of the pegylated nanoparticles and the nanosystems is quite low of the total IV administered dose. So after such a low targeting, how these systems there can be labeled as the magic bullets? Hence, there is still a far way to realize the full potential of the nanotherapies to cure the dreaded diseases in the global community. Despite all the advantages and the challenges which are faced by the current targeted approaches, it could be implied that the marriage of nanotechnology and the medicine has resulted in a progeny which exhibit the potential to revolutionize the future drug delivery science. So, there is an imperative requisite to optimize the integration of the nanomaterials into the drug delivery system on the basis of the standard metrics and the protocols. The designing of the nanotherapies should be based on a well-documented aspects of the biomedical field and the surface chemistry of the nanomaterials. With the emergence of the nanomedicine, a new area is also impacting the outcomes of the drug delivery, which is nanotoxicology by the biomaterials. In the light of these facts, the challenges posed by the nanomedicine should be given prior importance rather than searching for the new strategies for a drug delivery approach. Thank you.